Hello and welcome to Dr. Varsha Gaitondi's channel. Today, I am going to discuss about a brief journey of chickpea from orphan crop to elite crop. In the recent publications, chickpea is getting more popularity as it is grown for and focused for climate resilient agriculture by ICRISAT led scientists. As you know, the basic focus of ICRISAT is on two pulses, two oilseeds, and one cereal. Chickpea is one among them. Let's discuss some of the basic things about chickpea. Caesar aeratum or Caesar cabulian is the scientific or botanical name of chickpea. The production ranks third among pulses and cultivated in more than 50 countries, especially in South India and Sub Saharan Africa. It's an important source of protein, dietary fiber, micronutrients, key to nutritional security. The demand for chickpea is set to increase in the coming years as world population is rising already. Research like modern one are need of the hour to help the major chickpea producing countries like India to boost the production while making crops climate resilient. So more than 80,000 germplasm accessions are being conserved in 30 germplasms across the world, but only a few have been used for chickpea improvement. So as I was telling, there are two type of varieties or the basic kinds of chickpea, that is desi type and Kabuli type. Desi type is the common consumption in India, like we use it for basin or in our daily table purpose, and whereas Kabuli type, if we talk about or if we remember chola bhatura, Kabuli type chickpea is used the white and bold one. Desi type occupies more area, whereas Kabuli less. Color of the seed is yellow to dark brown, and for Kabuli it is white or pale cream. Desi type seeds or grains look smaller, whereas Kabuli are large, bold, and attractive. Shape of the seed is irregular and wrinkled in case of Desi, whereas Kabuli are smooth one. The yield potential of Desi type is about 2.2 tons per hectare, whereas it is lesser than Desi type in Kabuli type, that is 1.5 to 1.8 tons per hectare. The popular varieties of Desi type is Jyoti, Annigeri, JG11, Jaki 9218, Kranti, Bharati, and if Kabuli type means Shweta, KK2, and Vihar. Unit cost of production is lower for Desi type and higher in Kabuli, and price per kg is lower in Desi type and high in Kabuli. So overall, nowadays for table purpose, Desi into Kabuli type means the crossbreeds are coming to market, which are growing fastly and coming towards the markets before the seasons. Okay, so now we were discussing about the climate resilient chickpea, how it grown and how the how it is leading the world nutritional security. So here the major focus is on improving it for nutritional security, improving it for climate resilient. Climate resilient means it may be for disease, it may be for pest, it may be for biotic or abiotic stress. So the scientists of ICRISAT sequenced the genome and they used different modern approaches. They identified about 3,366 lines or accessions from 60 different countries. Of these, 3,171 were cultivated while 195 were wild species. Why I am talking about the accessions or the germplasm variants across the world? Because whenever we go for genomic studies or pangenomics or haplotyping, we should need the diverse number of genes. In one of the interviews of Rajiv Varshni, he was talking about increasing the good karmas of chickpea and purging the bad genes. There he was talking about the importance of wide number of accessions across the world. This also paves a way for the origin of chickpea when they went back to the origin of chickpea. As you know, agriculture and human settlement went hand in hand, and it took about 10,000 years above and confirmed that chickpea's origin was somewhere in Fertile Crescent and provides a complete picture of genetic variation within chickpea. The origin place gives complete variation where we can collect the more number of accessions. The Fertile Crescent is the swath of area in West Asia that is home of some of the world's earliest cultivation and civilizations. Here, about 1582 novel genes and established pan-genome of chickpea, which will serve as a foundation for breeding superior chickpea varieties with enhanced yield, resistant to drought, etc. The combination of artificial intelligence and fast tag is nothing but information technology with genomic prediction technology or DNA prediction technology to identify the combination of genes most likely to improve crop performance. Means from the orphan crop to elite crop journey, chickpea is having the contributions from different fields. It is not only from the breeding section but even the agronomic traits or agronomic practices were also improved. Means agronomist contributed like when to apply fertilizer, how much to apply, which time of sowing will be better for different varieties, and to which zone which variety is suitable. All these things along with the pathologist, entomologist and other people's contribution, maybe the physiologists and all. Along with that, IT technology, artificial intelligence technology, and the modern genomic tools contributed for this. So, when we basically focus on the genomics or breeding perspectives, let us see some of the key findings about chickpea origin. The analysis revealed that chickpeas, after originated in Fertile Crescent, migrated across the globe following two routes, one from South Asia and East Africa and the other to the Mediterranean region plus the Black Sea and Central Asia. Caesar reticulatum and Caesar echinocospermum were estimated to have diverged around 15.3 million years ago, which is higher than previous estimates and might be influenced by wild accessions conserved at Ikrisat represented only some populations of these species when recent work shown that Caesar echinocospermum populations are cross compatible with Caesar ariatinum. Introgression from Caesar echinocospermum into cultivated chickpea, which is widespread in Australia and Northern American breeding lines and is also likely to have occurred in international center, that is Icarda lines. That means the work is mainly focused on drought tolerance because the accessions of Caesar echinocospermum are somewhere resistant or tolerant to drought to some extent, along with other abiotic stress factors. So, after this study, chana is among the club of crops like rice, wheat, maize, for which there is extensive genetic information establishing future advances through applied genetic technology. That means now it is somewhere a light crop where it can be talked in the levels of rice, wheat, maize, which are the major or leading pulses across the leading cereals across the globe. So, why genomics or sequencing approach talked in any of the crop? As you know, Human Genome Project, which was a successful story, and because of that, we could identify many of the genetic disorders and diseases and could overcome or repair in the early stages of human development by using either genome editing or some other advanced medical technologies. Like in the same way, in plants also, sequencing is very important. What is sequencing? It is just the combinations or seeing the arrangement of ATGC or the nucleotide bases. Germplasm sequencing efforts in some crop plants have provided insights into the global distribution of genetic variation, how this diversity has been shaped by the genetic bottlenecks associated with domestication and by the efforts of selective breeding, how we can link this genetic variation to phenotypic diversity for breeding applications. These three key features are very much important for sequencing. The haplotype maps developed using whole genome sequencing data have helped to determine the percentage of constrained genome and detect deleted mutations that can be purged for accelerating breeding. Sequencing and genotyping the germplasm collection allows better conservation of management in gene banks. That means what we'll do in gene banks, we will be just taking and bulking or dumping all the genetic variants present with us. But just imagine how much cost as well as the human resource is diverted towards this issue. So, if we'll identify the good combinations, that means if we are increasing the good karmas of ours, we will be living peacefully in this world. Just like that, in case of crops, if we are purging the deleterious genes and only keeping the good genes or the haplotypes. What is haplotypic combination means? It is like supermarket where if we compare the normal shop and supermarket, in normal shop whatever the shopkeeper is giving that we are taking, but in supermarket we are having our own choices. That means 50 or 100 types of same products will be available with different labels or companies. So what we'll do, whichever product is suitable or we like, either it may be shampoo, whatever it may be, like that. In germplasm or gene banks also, we can take only the good genes. Good genes means gene of our interest. It may be for any trait, maybe for qualitative trait or for biotic or abiotic stress or for anything it may be. So, there are key features or key points for chickpea research to become popular, like studies based on chloroplast or ribosomal DNA. It was not only based on the simple DNA sequencing, but they also focused on chloroplast DNA as well as nuclear and ribosomal DNA. The two genomic breeding approaches like haplotype based breeding, just whatever I talked, haplotype is nothing but it is the combination of good genes where the nucleotide sequencing is made. Then genomic prediction. What is genomic prediction? We will take in the separate talk. Then OCS, that is optimal contribution selection. This also, what is optimal contribution selection, I'll discuss later. This helped in providing tailor-made high yielding and climate resilient varieties. Then they identified the candidate genes. Again, candidate genes means the genes under question or the genes which we want under domestication and breeding that were responsible for reducing genetic diversity in cultivated gene pool. Genetic load in incisor species. What is genetic load? It is the load of deleterious alleles or the deposition or contribution of deleterious alleles. If we decrease the genetic load in any crop, we can increase its traits. So, through selection and recombination, that is marker assisted backcross breeding or the conventional backcross breeding, were successfully purged many deleterious alleles. The current collection of breeding lines and cultivars still contain substantial genetic load that affect crop fitness. That means to increase the fitness, somehow we should breed further in order to reduce the deleterious alleles. There is a numerous superior haplotypes for improving related traits in land races. So this was the basic article about the improvement in chickpea genome I read, and it is quite good. That is chickpea genetic variation map based on the sequencing of 3366 genomes. That means 3366 lines they pulled at one place and they studied. This is really a very time consuming and very tough job where they collected the accessions from the origin, center of origin, as well as the depositories across the world centers. So here the key lines are constructed chickpea pangenome to describe genomic diversity across cultivated chickpea and its wild progenitor accessions. To show you pangenomics in very understanding way, what they will do, they'll go for Venn diagram. That means this is one accession and this is another accession, means there will be some common good traits in both. So what will happen, these two will overlap. For example, this is also having one high protein gene and this is having another high protein gene, both are different. So what will happen, they fall under this category. Like that pangenomics will work. A divergence tree using genes present in around 80% of the individuals in one species allowed us to estimate the divergence of sizer over last 21 years ago. That means to become a light crop, it didn't happen overnight or within few days. It took years together. Slowly the development happened and the consumption pattern of human also matter a lot here. There was continuous selection during domestication, migration, and improvement. That means selection and recombination paved a way for the basic improvement of crops. This particular crop. So again here three approaches are important in genomics. The chromosomal locations of deleterious mutations responsible for limited genetic diversity as described. Fitness were identified in allied germplasms. That means purging genes were identified in allied and removed or reduced in allied germplasms. Scientists identified superior haplotypes for improvement related traits in land races. Haplotype based breeding found target for purging deleterious alleles through genomic assisted breeding or gene editing. As you know, gene editing is a modern tool and quite successful nowadays. They proposed three crop breeding strategies based on genomic prediction to enhance about 16 traits. This I have already discussed three strategies. Along with that, the additional strategy is predicted performance of 100 seed weight, an important yield related trait increasing up to 23 and 12% with the OCS and haplotype based genomic approach. So about purging bad genes I have already discussed. So what we can do in future about chickpea improvement, there are few better places in the world for dryland agriculture research. The aim is to look forward to engage the local farmers, local industry, and the wider community to develop solutions to some of the highly complex problems we are facing in Asia and Africa. That means not only the breeder or agronomist or agriculturists or scientists, the combined approach of people and the government and the scientists participation is very much important in the improvement. So why I have put this phylogeny based clustering diagram, because many of the students won't be understanding what are all these coloring codes. So here you can see there are three circles. So 1 2 3 numbers are given here. First one is biological status. Like this is the first circle from inside. Here different colors are given. The colors blue means Desi type. See here many much quantity of blue is indicated here. Then intermediate between Desi and Kabuli. Then wild. Wild is with lavender color. This is the wild. Wild is also occupying very less area of germplasm accessions. Second one is market class like breeding lines, cultivars, land races, and wild forms. Before getting into any discussions, you should understand some of the basic genetic concepts. I hope you know them. So the second circle is about market class, this one, and the third one is geographical origin. That means green is American, then red is black sea. Here you can see all those things. If you zoom it and scan properly, you will get the colors like see the black sea red color is indicated here. Likewise, you can read the phylogeny based clustering. This clustering is shown as I discussed about the 3000 plus accessions which were collected worldwide, and they were presented in this way to understand them in a better way. As I promised you, I will discuss the terminologies. Here it goes. Haplotype means group of genes within an organism that was inherited together from a single parent. Haplotype word is derived from word haploid, which describes cells with only one set of chromosomes, and from the word genotype, which refers to the genetic makeup of an organism. Haplotypic map, small, shortly or smartly called as HapMap, a tool that allows researchers to find genes and genetic variation that affect health and disease. Phenomics, as you know, it is a branch of omics. Change is seen in an organism resulting in the phenotype. Phenotype means the external appearance during the lifespan. Next is pangenomics. Pan or supragenome is the entire set of genes from all strains within a clade. Genomic prediction, identifying the regions of genomic DNA that encode genes. OCS is a balance between rate of inbreeding and genetic gain. This selection process maximizes genetic gain in the next generation while constraining the rate of inbreeding via restriction of relatedness among the offsprings. Next, I will just focus on or throw a light on omics technology. As you know, omics is measure of some characters of a large family of cellular molecules like genes, proteins, or small metabolites have been named by appending the suffix omics in genomics. The technology includes genomics, that is study of genes and their function, proteomics, study of proteins, metabolomics, study of molecules involved in cellular metabolism, transcriptomics, study of mRNA, glycomics, study of cellular carbohydrates, lipomics, that is study of cellular lipids. Omic technology provides tools needed to look at the differences in DNA, RNA, protein, and other cellular molecules between species and among individual species. This is a diagram. Recently, in 2017-18, you might have heard about the Super Anigeri chickpea variety. This was improved by the contributions of US Raichur, University of Agriculture Sciences Raichur, and ICRISAT led team, and it was popular because of its character towards fusarium wilt resistance as well as to some extent drought and yield. So here I have given the diagram how the genomic assisted backcross breeding contributed for improving the particular lines. So if you are interested, go through it. Then some of the characters of Super Annigiri 1, which is released for Andhra Pradesh, Karnataka, Maharashtra, and Gujarat states. Now the wide production for distribution to farmers efforts are going on. This developed, as I told, for fusarium wilt from WR315 genotype of chickpea in the genetic background of Annigiri 1, an old but leading chickpea variety of Karnataka state for many decades. Superanigari 1 variety has an average grain yield of 1,898 kg per hectare and has recorded about 7% increase in yield over Anigari 1 and is highly resistant to fusarium wilt, an important yield reducing factor in South India. Variety has an average maturity period of 95 to 110 days and is semi-erect plant type and attractive seed size of 18 to 20 grams per 100 seeds. Erect means it can tolerate to high temperature as well as more photosynthetic ability. This is all about how the genomics contributed for the improvement of the crop, along with that how it originated, where all it traveled, how Kabuli came all the way from Afghanistan to India, along with that what are the modern approaches like it may be IT technology, artificial intelligence, genomics, haplotype based breeding, pangenomics, and the combinations of omic technology helped to bring the orphan crop to a light crop and attracted the world people's attention. That's all I discussed in today's discussion. If you have any doubt, you put it in the comment section. Always your suggestions are valued and expected. Thank you once again.